So when we first started hearing about COVID-19, we were concerned. Even before March, we had already launched our first call for proposals in our community to activate them because we really did think the writing was on the wall and that this virus was coming to Canada. We didn't have any idea how big it would be when it got here, but we did think we were going to have to deal with it, and we wanted our community to be ready. While the world was shutting down in March, we were gearing up. And we called on our community to send in their ideas that could have short-term, immediate, meaningful impact in about a 6-month time frame. And we activated the top projects right away. We received 173 applications that were all reviewed within a couple of weeks. We selected the top 67 out of all of those projects and funded them. And they have gone on to make incredible contributions in clinical trials, new therapeutics, new diagnostics, in public health measures, in helping support the communities and understanding the effect of public health measures on mental health, education, and family dynamics. We've seen the power of science in this pandemic. Things like the clinical trial platform we supported, the level 3 hot lab where researchers could work safely with COVID-19. And those are just the obvious science ones. And many of our community members have been leaders in translating the science and the public health messages to the community in a way that people can understand. When you talk about a pandemic, when you talk about infectious diseases, they are completely interwoven with many facets of our lives. Think about the knock-on effects from public health measures on education, on mental health, on the economy. These are complex issues. You find the answers to how to deal with these problems when you have a diverse group of experts who look at everything with different lenses. These are the people you need to address the type of complex problems that we've seen with COVID-19 and the type of problems that we are still facing in the community going out of the pandemic. I'm very, very happy with what we've accomplished. But not time to rest on our laurels. We initially had a long list of challenges that we wanted to face. Things like immune-related diseases, using the immune system to cure cancer, tackling the antibiotic resistance crisis and the loss of antibiotics, which threatens our whole healthcare system again in a far worse way than COVID-19 did. These are the fundamental challenges that MI4 is re-engaging with right now, so that as the pandemic starts to wind down, we wind up.
My name is Nicole Basta. I'm an associate professor in epidemiology at McGill University, and my field of research is infectious diseases and the impact of vaccines. My research really focuses on how we can prevent and control infectious diseases. So even long before COVID-19, we were working on a number of diseases that really have a global impact. So what we're trying to do is figure out what is the best way that we can ensure that people stay healthy so that they can enjoy their lives as fully as possible. So our research is focusing on the population level, trying to figure out what those strategies are that can ensure that infectious diseases don't cause a great deal of disruption to health and society. Early on in the COVID-19 pandemic, since I was an infectious disease epidemiologist, I was getting so many questions about when we would have a COVID-19 vaccine. I think a lot of people had a lot of hope that a COVID-19 vaccine would have a great impact on preventing and controlling the pandemic. So my team realized that with all the different vaccines that were being developed and all of the different clinical trial evidence that was coming out, it was creating a lot of confusion for the public. So we developed the COVID-19 vaccine tracker to try to make sense of all of that information. So it's a single resource that brings together all of the evidence and all of the data about all of the different COVID-19 vaccines that are being developed around the world. We also track all of the vaccine approvals now that several different vaccines have been approved, so that people can find out which vaccines are available in which countries and which countries have begun vaccinating with COVID-19 vaccines. So we really found that there was a great need to try to simplify the information about COVID-19 vaccines for the general public and to provide a resource so that people had a one-stop shop where they could find out about scientific progress in terms of the development of COVID-19 vaccines. I work with Professor Erica Moody and a fantastic team of students, trainees, and other researchers. And without their support, we really wouldn't be able to make this resource happen. Our COVID-19 vaccine tracker is a daily undertaking because we're sorting through so much evidence and so many news reports and press releases about all of the COVID-19 vaccine advancements. We really have to use our expertise and drill down to look at all that evidence and consolidate it for the general public. So having a strong team working on this resource has just been an incredible asset since our early days. So far, we've had about 11 million page views on our COVID-19 vaccine tracker and 3.5 million users around the world from a whole diversity of countries. So we are really proud that we've been able to use the support from MI4 to create this resource so that anyone, anywhere can access information about where COVID-19 vaccines are being developed and where they're currently available.
Okay. Thank you, everybody here, for joining us this evening. And those of you who are on video, thank you for joining us in the virtual space. Good evening et bonsoir tout le monde. My name is Marcel Baer, and for a few days now, I've been the interim director of MI4, which is really exciting. It's an incredibly steep learning curve. And this evening, it's a great pleasure to welcome you to the official launch and the keynote lecture of Infection and Immunity Week, which is part of the McGill Bicentennial. This week of symposia about infection and immunity gives us a chance to reflect, to celebrate the past, to reflect on what makes McGill great today, and also to ponder where and how McGill can contribute in the future. As an example of where the past and the present intersect, I want to take a moment to reflect that McGill University is on land that long served as a site of meeting exchange amongst Indigenous peoples. The full land acknowledgement is projected behind me. I'd like to point out in 2021, the Indigenous peoples of Canada suffer disproportionately from a number of infectious diseases, including tuberculosis, the disease that I study. And the work that we do at McGill in the infection immunity space needs to be responsive to the people in our communities, and for those of us in the hospital-based faculty, to the people in our RUIs. So as we ponder the future, let us be sure to consider the persons we work with in the communities where they live, to make every effort to include these people in our plans as partners and collaborators in the scientific and the medical journeys ahead. At this point, it gives me great pleasure to call upon our Principal and Vice Chancellor, Suzanne Fortier, to say a few opening remarks. Thank you.
Bonsoir à vous tous et bienvenue. C'est vraiment un plaisir d'être ensemble une fois de plus, là, ici au Club de la Faculté. And I must say it's really wonderful to finally be able to meet in person. I know that many are joining also via the Zoom, and so welcome to you as well. As Marcel said, it is the launch of the Immunity and Infectious Week at McGill. Infection and Immunity Week at McGill, and it is one of the very first events of the bicentennial that we can have in person. So we're very, very happy. Gérard Cadet in particular is extremely happy that we can be here in person. One small point, but very important one about the bicentennial events is that many are focused on what our friends and alumni really want from McGill, and it is good, reliable knowledge. And we had one on the problem of food security at the beginning, and now we have this one. So I can tell you that all of the friends of McGill will be watching this event. C'est évidemment une belle occasion d'être ensemble pour célébrer la semaine là, qui est en train de débuter. Et puis, on s'attend évidemment à ce que vous ayez des conversations très fructueuses, qu'on puisse se parler de ce qui nous intéresse tous et qui intéresse vraiment la planète au complet. Et j'aimerais aussi profiter de cette occasion pour faire quelques remerciements. In particular, I want to recognize the generous donors that made it possible for MI4 to become one of the largest group of experts focused on infectious and immune-mediated disease in the world. Et on sait que beaucoup d'entre vous, du groupe de MI4, sont ici ce soir. Alors, on vous dit un gros merci. Évidemment, we thank you for all the work you did very early on. You were on it, as they say, and you were ready to put all of your talents and expertise and efforts to work so that we could, not only in Canada, but across the world, be safe. You also had another impact. You may know that, you may not. When we do surveys of the appreciation of the general population for universities in Canada, we've seen an increase. And that's largely due to you that we see the increase of appreciation from the general population because they really appreciate you sharing your knowledge. And one place we've seen it also outside of Canada is in the US. Very, very big increase in the appreciation of the general population for the advanced research universities in their country. And je fais un clin d'œil ou un petit signe ici à notre invité qui va nous faire une présentation ce soir, because I have to say Johns Hopkins was a big contributor to the increase of the appreciation of Americans for their research-intensive university. Of course, you all know that the pandemic has really put a highlight on your work, has shown the whole world the importance of what we do, and I think that it will continue to be the case that many people now want to support the work that you do and will be helping us in reaching additional initiatives and being able to launch new initiatives. Alors, je vous remercie beaucoup, vraiment, les chercheurs, les professionnels de la santé, pour tout le travail que vous avez fait. The whole world is grateful for what you do, and I think that you should feel a sense of real a recognition for your work, but also a sense of achievement, because you have really carved an important place in helping us fight this terrible pandemic. And we know now that infection and immunity are very, very important areas of research. I'll tell you a little story to end, because it was at a talk with Don Shepard. He was meeting with alumni, and the thing he shared with us, which most of us could not believe, it was that over 75% of people died from infectious disease. And it struck us as a bit of a high number until he explained that many times people have a condition that makes them vulnerable, such as cancer or other cardiac problems and so on. And so these people are less able to fight any infections. So although people will normally say, well, they died of cancer, in fact, what really was the cause of their death was the infectious disease that they were not able, their immune system was not able to fight. And so I think it was a big eye-opener, certainly for many of us who were there. But again, with the pandemic, we saw that indeed those who are vulnerable and have immune systems that are vulnerable are ones that are most at risk in these situations. So again, I think that people have a more acute sense of the importance of what you do, but also an acute sense of the acknowledgement and the gratitude we have for all people who are putting their talent and their expertise for our health. So, alors, c'est peut-être vous participez à l'échelle mondiale à notre bonne santé. Alors, je vais faire un toast en mode symbolique à vous tous pour nous souhaiter à tous bonne santé. Merci.
Thank you, Principal Fortier. And I'll get my little clicker to advance the slides. So first, I'm just going to tell you a little bit about the week. Obviously, we are thrilled to have been chosen as one of the flagship events of the McGill Bicentennial. And of course, having this many events in this many days is both it's an honor and it's a great pleasure. In some ways, it's like playing for the Habs, but more like last July and then this November. So here's an overview. This morning, there was a symposium that was jointly held with the Helmholtz Institute on looking towards personalized infection medicine. It was very exciting to have Ugur Sahin from BioNTech give the keynote and tell us about the development of mRNA vaccines, and then to have 3 McGill investigators. Don Vin is one of them. Is Alexandra here? Yeah, Don is there, Alexandra, and please help me who the third speaker was. Patricia. But Patricia's not here, right? I'm looking for the speakers. So we had 3 from McGill and 3 from Germany, and it was really a wonderful conversation and different perspectives on what person-centered and personalized medicine can be in the future. This evening, we're going to have a keynote lecture by Arturo Casadevall, and soon Don Shepard's going to introduce him, talking about just the big concept. What is virulence? What's the origin of microbial virulence? And how does that shape what are the species that are on the planet today? Tomorrow there's going to be a session on One Health, defining actions to face emerging zoonotic infections. I believe Cedric Yansuni here is part of this effort, and we look forward to that session. There's a session on New Frontiers in Infectious Inflammatory Diseases. Sylvia Vidal and Ciro Piccirillo, among others here this evening, have organized the Wednesday session. We're going to hear about antimicrobial resistance and the microbiome. We have Ira King from the microbiome, and Dao Nguyen has arrived, there you are, who has just recently launched the Antimicrobial Resistance Center. And then on Friday, we're going to have a student day. And that's going to be really exciting because the students have made the agenda and they've created the curriculum, and it's up to us as the faculty just to sit back and enjoy it and to learn from it and to hear their perspectives on where infection immunity is. Here are the partners for the week. So for this morning, it was the Helmholtz Center, the J.D. McLean Center will be involved, the Center for Viral Diseases, Chen where are you right now? There you are. Chen Lang, who just created that, is a partner, and then Complex Traits, Antimicrobial Resistance, SETI, CHIROS, the Center of Excellence of Translational Immunology, the Microbiome Center, and the Graduate Student Association are our partners. And then there's many people behind the scenes who've organized this week, and Marie's going to thank them and remind everybody at the end how many people have contributed to making this week happen. So, I've been asked to give you a little bit of the do's and the don'ts. The slide looks like it's the don'ts on the left and the do's on the right. So if you're going to take selfies tonight, make sure you're wearing a mask. And if you're going to take pictures without a mask, quarantine the pictures for 14 days before you post them on social media, because if there's one person in the room who has COVID, we don't want to know about it 2 days later. Speaking of people with COVID in the room, Jesse Shapiro reminded me that the Trottier Center actually has an asymptomatic screening platform, and I went by there this morning, and I'd just like to give a shout out to the volunteers there. Victor and Sophie, who tested me and told me that I'm negative and gave me just that extra little bit of confidence to come into this room, and I think that's just another great initiative on campus. We're always looking for new initiatives and new ideas and new directions. To make 2022 even better than 2021. You've been given some notepads at every table, so speak with your colleagues and see what kind of ideas you have, scribble them down, share them with us. The best ideas, the craziest ideas. We'd like to hear what you think MI4 can do next year, and just share those ideas with us. This evening there's going to be a really amazing keynote talk. I know it's amazing because I got a sneak view on the weekend. And so we look forward to hearing the talk, and we also invite you to mingle afterwards and talk in as safe a manner as possible. At this point, I have the honor of introducing my colleague, my accomplice, and my friend, the founding director of MI4, Dr. Donald Shepard. He's not here in person, but he's here virtually, and hopefully in some sort of a choreography like Cirque du Soleil, he's going to just show up magically on the screen and take over from me. Kind of like I throw and he catches, but I'm not sure when that's going to happen. Don, as many of you know, but some may not know, is now working at the Public Health Agency of Canada, where he has a role in working with and leading antimicrobial resistance efforts. We here all wish him the best in his new position, but we're delighted that he was able to join us this evening. He's going to be introducing his fellow mycologists. Now, in mycology, they call each other fun guys, so that's sort of the joke. So one fun guy is going to introduce another fun guy, Arturo Casadevall. Don, the virtual podium is yours. Hopefully you're there.
Marcel, can you hear me? To the stage so that I know I'm on. Anybody? I'm live.
I can hear you.
You and I are on. I'm not sure
Right.
if I'm on.
I don't see you on the screen.
I don't see myself on the screen, so that's not a good sign. Oh, I see the room. Can you hear me, folks? Yeah. Marcel, ask you for those thumbs. All right, this is an unbelievable to introduce our, and not just because he's important organisms on planet I, but because he is a fixture in my not just mentorship, generation as to what actually do if one is a scientist and an excellent thinker. Actually, on paper, Casadevall is the Bush professor at Johns Hopkins School of Public Health and School of Medicine. He is the Alfred and Jill Sir and chair of the Doug Feinstone Department of Biology and Immunology. Is a world clinician scientist, superb researcher, very deep thinker who's specialized in how anti-host defense and antigens, a word that he will take issue with, and in fact, what antigen. He's really minds in modern energy. He challenges, he brings big forefront, although he's always forced to hide the miracles, he tells me. Hey, you're going to be privileged to go whirlwind tour. Examine our relationship with monkeys, weaving a fascinating, begin in the age of the dinosaurs, walk through the meteor, the great extinction event. look at the rise of modern, it's going to look at our present life, and it's just a little bit of climate change for the next 4 years. I think I'm, so I will stop, pause, and hand it over.
Good.
Am I back on?
Hear you, but this thing is pretty real. I guess somebody will. Thank you, by the way, for that introduction.
Off my video, not because I asked by it, but simply to quality of the
Right.
You deserve every far more.
Very. There we go. Thank you for the invitation to do the keynote address on the celebration, on the 200th year celebration of McGill. I think of institutions as almost like living entities. And when you think about 200 years, you think about all that came before you, all the people that struggle in departments, taking care of patients, teaching students, doing research, and all of that. And it is a great accomplishment to have maintained that continuity and that continuity with the past. In fact, some of my talk will begin discussing events that happened when McGill was already well established, the germ theory disease. And I'm looking forward to sharing with you some thoughts about the origin of virulence. So thank you, and thank you for including me in your celebration. The title of my presentation is on the origin of microbial virulence, and it's going to draw strongly on insights from the fungal kingdom. I'm very interested in history, and I think that a talk on virulence should begin with the history. So the germ theory disease was one of those landmark moments in human history when everything changed. Physicians made an association between microbes and certain diseases, and that allowed them to be treated, vaccines to be developed, sanitation to be enhanced. And one of the fascinating things about this time is that one of the giants of the 19th century, Virchow, did not initially agree with it. And part of the reason was he was very interested in social issues, and he worried that if microbes were associated with disease, there'd be less emphasis on taking care of poverty. I say that to you because we live on what appears to be a divisive time. People are very divided in many ways. But it is important to recognize that that even great minds can be divided, and that one can work together and find a way forward. I also want to recognize other heroes that should be on stamps that are often not included. Women and people from underrepresented groups, and I'm going to acknowledge some of their contributions. Fanny Hess discovered agar, which is very important in microbiology. Anna Williams, the diphtheria vaccine, a major killer of children. Marjorie Stevenson isolated the first enzyme from bacteria. Today, we take that for granted, but that was huge. It established that the biochemistry of bacteria and other organisms was continuous. Ruth Ella Moore was the first African-American PhD in microbiology in the United States and a pioneer on tuberculosis therapy. And Jesse Price developed a vaccine against Doc Pasteurella. The germ theory disease had 3 requirements. You needed a new technology, the microscope. It allowed people to see microbes. It required observation and description. It is, for example, the broad street pump, the association between childhood puerperal fever and hand washing. And it requires some experimentation. It did not require mechanism. Mechanism is something that comes later on the 20th century. One of the things that is forgotten is that the fungi led the way on this. Agostino Bassi made the association with saltworm disease and a fungus that is named after him. And David Gruby associated ringworm disease with a fungus, and it was the first human pathogen associated with disease. So when you think about mature scientific fields, one could ask the question, where is microbial pathogenesis? We are healthy in observation. We are healthy on experimentation. We are weak on theory. For example, the theories can be stated in some single sentences. In the 1890s, people thought, oh, microbes cause all diseases. By the 1920s, some microbes cause some diseases. By the 1970s, when the problem of opportunistic infections was first realized, some microbes cause some disease in some hosts. When it comes to prediction, we are poor to none. And I point out that other theories today in other fields like geology has plate tectonics, biology, evolution, chemistry, quantum physics, and cosmology, general relativity. And this is an area that I think that we need to pay more attention to. So one of the things that we developed was the damage framework of microbial pathogenesis, and I'm going to share that with you. This came from teaching. It turns out that Dr. Lysiane Porowski and I used to teach a microbial pathogenesis course, and we had a lot of trouble teaching the students without providing a comprehensive framework. But we also realized that there was a late 20th century crisis. So in 1900, there were two types of microbes, pathogens and non-pathogens. By the 1950s, non-pathogens began to be associated with disease in the setting of steroid use and the advancements of the 1950s, antimicrobial use and early chemotherapy. That led to the concept of opportunism, to try to explain how non-pathogens cause disease. But by 1990, a walk on a hospital would reveal that most of the organisms being recovered from patients would have been non-pathogenic at the beginning of the 20th century. For example, Staphylococcus epidermidis, Candida albicans. So the question is, what happened? And what happened was that the host had changed, and microbe-centric theories of pathogenesis could not account for those changes. So the crisis led to a messy lexicon. For example, Candida albicans is a commensal in asymptomatic humans, a primary pathogen in vaginal candidiasis. There is nothing wrong with those women who get this, or an opportunistic pathogen in AIDS. In fact, you could be the Candida albicans can be in all three states in the same individual. When you ever have a messy lexicon, you know that there are intellectual problems in a field. So as I pointed out, we were involved in teaching, and we developed this concept of the damage framework that came from teaching microbial pathogenesis. What happened is that we realized that our class notes had new thought. And the question is, what do you do with it? Where do you publish new ideas today? We are very focused on experimental science, and it's very difficult to publish thought. So what we did is we hid it in a mini review. I think that continues to be a venue by which you could do so. And this turned out to be today one of our most cited papers. So we know that thought and theory are not highly valued in the current biological sciences. The only way to get this published was to hide it inside of a review. It's very simple. It argues that every host microbe interaction is some derivative from a parabola. And that this parabola, on the x-axis, you have the host response. And we are very qualitative, go from weak to strong. In the y-axis, you have host damage. So, and what matters to us is in this interaction is host damage. Disease only occurs when sufficient host damage has accumulated to interfere with homeostasis. But what is novel about it is that it sees all these states as continuous: commensalism, colonization, latency, disease. They differ only by the amount of damage. And if you drag that curve below the x-axis, you can see beneficial interactions, symbiosis. The damage framework had 5 consequences. It shifted the base from pathogen and non-pathogens to the outcome of the interaction. In our view, there are no pathogens, even though I know we can't give up the word. No commensals, no opportunists, no sacrifice. There are only microbes and hosts, and when you put them together, you get a new state. And that state can be disease, commensalism, symbiosis, etc. These outcomes are continuous and differ only in the amount of damage incurred by the host, and it sees virulence as an emergent property and I will return to that at the end. So again, in 2015, we added the microbiome, but note that we are still inputting it in many reviews. This, for example, is very adaptable. We were able to adapt it to explain some aspects of the pathogenesis of COVID-19 last year. And you could see where people are in presenting disease, decompensated disease, why we use steroids to move individuals, try to move them around the curve to reduce their damage. The bottom line, I think, is that this is very adaptable, and it gives a different way to approach the problem where you take the contributions of both the host and the microbe. Raises the question, what is a host? We were surprised that we couldn't find a definition of a host in the literature. How can that be? We take it for granted. Well, it turns out that a host is a chair a host, is a rock a host? The answer is no. But a fly is, a worm is, a tree is, and a human is. And the difference is that these entities can respond to the associated microbiome. The rock has a microbiome, and so does a chair, but it stays static. Whereas a true host is able to respond with their immune system and create a brand new state that can be symbiosis, colonization, commensalism, latency, or disease. So I've given this lecture. The editor of Nature asked me to address the issue of the lexicon. And in this article, which is quite provocative just from seeing the word, we argue that the problem is that when you call a microbe a pathogen, you're giving it a trait that is not its own. No microbe can be a pathogen without a host. And what we argue is that since the way we think uses words, we use symbolic language. If our terms are not correct, we may not be designing the best experiments. I don't expect everyone to give up the word pathogen or commensal. I just hope that people think through and realize the limitations in the terminology that is used. One of the things that I was interested in was, could a mathematical formalism be developed that would account for this? In other words, could this be worked out in some sort of math? And after a lot of thinking about this for several years, developed the concept of the pathogenic potential of a microbe. And playing around with terms, it is defined as the fraction symptomatic over the inoculum multiplied by an amplifier term where you take 10 to the fraction that is mortality. If none of the hosts died, this n becomes 0 and this term becomes 1. But if hosts die, well, host is a pretty extreme event, so that becomes an amplifier. And then I went into the literature and I looked at the mouse experiments, and I was able to compute the pathogenic potential. Is very simple for many different types of organisms. And the important result that is continuous. You don't have a cutoff where you say, whoa, these are non-pathogenic. These are pathogenic. The take-home message is any microbe has some pathogenic potential and can overcome a host if given in sufficient amount. Now, you can change the host. So here you have, for example, 4 organisms of fungus, bacteria, 2 bacteria and a virus. And what you can see is that in mice, if you change the mouse strain, the pathogenic potential changes by as much as 100-fold. It incorporates the contributions of both the host and the microbe. It's beginning to be used, which makes me happy, in part because it is a better term than virulence. Virulence cannot be quantified. You always have to measure virulence relative to something else, a mutant or so. In this case, you can quantify it provided you know your inoculum, you know your fraction symptomatic, and if you have mortality, you record that. However, I realized recently, actually, while I was driving and thinking about this, that there was a problem. It was incomplete, that I needed to add the variable of time. And the problem is that if you don't add the variable of time, you could end up with the same pathogenic potential for 2 organisms that kill at very different times. So very simply, just you can change it to dividing by how long it takes for, in this case, the organisms to die. This paper is submitted and it allows you to compare two microbes where everybody dies, but they differ in how long it takes for death to occur. So now let's move to the fungi, because they teach us new lessons in the problem of virulence. The fungal kingdom is thought to be the largest kingdom by the old meaning of the word kingdom, the number of species. It includes major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Currently, it is devastating bats in North America. Fungi are causing catastrophic declines of amphibians. The salamanders in Europe are threatened, the snakes in North America. Yet mammals are remarkably resistant. Humans that are not immunocompromised don't worry about fungal diseases. People in this audience may worry about viruses, parasites like malaria, bacteria like tuberculosis. People don't tend to worry about fungal diseases. And to me, that's a fascinating aspect of the mammals, because certainly they are the major pathogens for a variety of other types of hosts. Not only do we not worry about them, but we have relatively few fungal species that are pathogenic for humans. Host associated, we have the Candida species, the dermatophytes, and pneumocystis. Disease occurs only when you disrupt the host microbe relationship. In the environment, there are a few others. You tend to either have to be immunocompromised or get a very large inoculum. So what are the requirements for fungal human pathogenicity? Well, to cause disease in deep tissues, the fungus must be thermotolerant. It must be able to grow at human temperatures. I'm going to show you data that the majority of fungal species cannot meet this test. And then you need to have, the fungus needs to have the capacity to survive in the host. It must have the so-called virulence factors. It must find a solution to evading the immune system. And there are many solutions. Some use capsules, others use toxins. You have antioxidant systems to prevent killing, intracellular replication, etc. I'm looking at this from the big picture. So let's ask the question, why mammals? Why are we the dominant large animals in the planet today? Could the fungi have been involved? Could the fact that we don't fear the fungi be in some way related to this phenomenon? So the origin of mammals goes back to the Permian. We have ancestors long, long ago. And at the time, mammals are thought to have descended from organisms known as cynodonts. And as late as the late Cretaceous, mammals were going nowhere. They were small creatures about the size of a squirrel, living in the margins of what was a reptilian world. You have to ask the question, what advantages do you have by being warm? Well, you have to eat a lot, but it gives you a stable internal environment. Your enzymes can function at one temperature. One has the ability to move and digest in cool weather, and perhaps protection against infectious diseases. But these advantages were not sufficient to compete with a reptilian. The Cretaceous world was a reptilian world. And I want to point out that the greatest extinction in the geologic history is the Permian extinction. And this wiped out 95% of all life. It is interesting that separating the Permian from the subsequent layers is a 1 meter layer of fungal spores. People think that this was the result of all the decomposition that went on. I would argue that anything that made it through this boundary would have had to deal with the fungi. In fact, there are fossils from the Permian that resemble mother plant pathogenic fungi, suggesting that the capacity for fungal virulence was found in deep time. The mammalian lifestyle, and this is what makes it so interesting, is it's very expensive. I already had 2 meals. I'm planning to have another one. And probably by the end of the day, I would have eaten 4 or 5 times. If we were reptilian, we could eat once a week. Mammals were a very minor class of organisms until 65 million years ago. And then there was the KT event, and I'll say a few words about it. But the question is, how did these organisms become dominant? We know that reptiles also survive. So the Paleogene fauna, this is the fauna after the KT event, is characterized by the great mammalian radiation. We went from those creatures that were small, squirrel-like, to the whole diversity of mammals that ranges from whales to bats to humans. And I asked the question, what could have selected for this energetically costly lifestyle? The great mammalian radiation was a tertiary event. So what you see there is that the dinosaur lines end. The birds make it through the calamity. Crocodiles make it through the calamity, and mammals just take off. What you see is the replacement of the reptilian megafauna with the mammalian megafauna. The Cretaceous world was a very different world than our own. It was a hot, warm, forested planet. The average temperature was about 10 degrees Celsius, hotter than it is today. The Atlantic was tiny, and this world came to an end with an impact in the Yucatan. And the rim of the crater is denoted by the cenote rings. If you go to Cancun, they offer you trips to cenotes. Well, that is the ring of the crater. Cenotes are found only in the Yucatan, and that represents the rapid melting and cooling of the rocks in the area. The post-impact world was a terrible world. The Earth went from being one type of environment to be totally devastated in a matter of hours. Giant tsunamis swept through North America and other parts of the world. The enormous forests were knocked down by the blast, and a lot of Earth got kicked up into low orbit that then re-entered as flaming meteorites. The amount of smoke, dust obscured the sun and shut down photosynthesis for at least 6 months, maybe as long as 10 years. Global temperatures dropped in a matter of hours by as much as 10 to 12 degrees. And there is evidence for massive fungal proliferation of the KT boundary, just like in the Permian. By the way, the Permian was not caused by a meteorite. It was probably caused by climate change as a result of coal beds having caught fire, which brings a warning and an echo to our time. But so here you have massive fungal proliferation. The fungal proliferation would have given you lots of spores. Lots of spores will have given you large inoculum. And remember the pathogenic potential equation. Every microbe has some pathogenic potential. So if you're exposed to very, very large inoculum, even low pathogenicity organisms could have caused disease. So I proposed this about 15 years ago, and it's simple. The post-impact world, the reptilian megafauna is killed by the impact blast, disruption of food sources, the chilling of the planet. Maybe some survivors would have been brought down by fungal diseases, given the massive numbers of spores. Reptiles fight off fungal infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases from the outside. In fact, one can find dinosaur eggs today that have been invaded by fungi. Eggs must breathe, they have holes. Hypha find their way into the eggs. And today, for example, Fusarium is a major pathogen of turtles. So in the post-impact world, the temperatures drop. The ectotherms cannot move or digest their food. They basically are too cold to move or to use their digestive system. It turns out that temperature has a major effect on reptilian sex ratios. So now whatever eggs survive, the sex ratios are altered, and that is going to cause more mayhem for their ability to repopulate a devastated world. In contrast, the mammals would have been able to move and forage because of their endothermy, and also their temperatures would have been able to keep up certain fungi. So the proposal is that a fungal filter at the end of the Cretaceous select for endothermy and mammals. Now, that brings, so the reason that we don't fear fungal diseases and that we are so resistant to them is in fact because we were selected by a fungal filter. So the fungal selection mammalian, I gave it a name, everything needs to have a name, call it FIMS, is very simple. After the KT event, you have both mammals and primitive mammals and reptiles survive. But the reptiles are greatly disadvantaged by the conditions in the post-KT environment. And that includes the probable susceptibility to fungal diseases given the massive numbers of spores. The net result was smaller founder populations. When the world began to recover, there were more small mammals than there were small reptiles. And therefore, that led to the great mammalian radiation. The mammals now outcompeted the reptiles, and you had the mammals as the dominant fauna. So this is, you can read about it. I update it every couple of years. This was updated just last year, I believe. What is the evidence that endothermy protects against fungal diseases? Well, it turns out that most of the organisms today that are vulnerable to fungal diseases are ectothermic, from corals to frogs to plants to salamanders to turtles. Organisms that cannot regulate their temperature above the environment tend to be susceptible to fungal diseases. In us, it turns out that it's our cooler parts of the body where we have, and for fungal diseases are common. Nail fungus, athlete's foot, dandruff in babies, fungal diaper rash. It's the cooler areas where you see problems in mammals. Experiments have been done that clearly show this phenomenon. John Perfect and others have shown let's take a bunny, a rabbit, and infect it with Cryptococcus neoformans. Well, you can't systemically. The rabbit temperature is about 40 to 41 degrees and basically prevents the infection. You can, however, give an infection in the cooler areas of the body. But here is a fascinating aspect. If you give steroids, then temperature is not enough to keep it, and you have a dead bunny. Take-home message is that it is the combination of adaptive immunity and endothermy that gives you the resistance to fungal diseases. The white nose syndrome in North American bats is an excellent example of this. It's a disease that was unknown to 2006, but now has spread throughout the United States and Canada. And what happens is the bats are immune in the summer. Their temperature is 37 degrees, and they can keep the fungus out. In the winter, their temperature drops to 10 to 12 degrees, and they become susceptible. And this white nose that you see is the high fall growth in the nose. If you take affected individuals and you bring them into the laboratory and feed them, and the feeding will raise their temperature, and that is enough to clear the fungal infection. Some work that we did long ago showed that most fungal diseases were in fact, that most fungi are not able to grow at mammalian temperatures. This was work that was done with Vincent Robert in a collection in the Netherlands. And when fungi are deposited in the collection, they always do a temperature susceptibility. So looking at about 4,000 fungal species, they do very well up to about 30 degrees. But then when you get between 30 and 45 and 42 degrees, it's a linear relationship where every degree allows you to keep off 6% of fungal species. That's a tremendous amount of protection from being hot. So I discussed this with my friend Aviv Bergman. He's a physicist, and this was a hallway conversation. And what he did, he went back into his office, he pulled out the equation of the cost. What is the cost of being hot, of temperature? That's given by a well-known equation. And what is the benefit of keeping the fungi out? And he solved the two simultaneously and asked, where is the maxima? The maxima turns out to be our temperature. This suggests, look, this could be a coincidence, but it also suggests that there may be something to this, that the reason that we are 37 degrees is because we're selected to be optimally protected to the fungi without necessarily having to eat all day. So the fungal infection mammalian selection theory has a lot of explanatory power. It's internally consistent. I've had no obvious, I can find no obvious contradictions or no significant pushback. It provides a selection mechanism for an energetic, costly lifestyle. It explains why nature ended up with the large animals being mammals that are hotter that have such a a high temperature. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It poses endothermy as a major host defense mechanism. And it explains the minor role for fever as a protective mechanism in modernity. Look, if you go into the hospital today with an infectious disease, you're likely to be threatened by an organism that is already temperature adapted, something that you caught from another human, or there is a nosocomial pathogen in the hospital. However, if one goes out into the wild and falls and gets dirt into a wound, then that organisms are not temperature adapted. So today, fever doesn't help us very much, but a fever against an infection caught in the environment could be life-saving. So the two great pillars that protect us against the fungi are advanced immunity. And we saw in the late 20th century how AIDS and the progress of medicine weakened the immune system, and those people become susceptible to fungal diseases. And the second thing is endothermy. Now, we don't have any condition where we can survive for a long time being colder. But what I would argue to you is that global warming may threaten the second pillar. So the world is getting warmer and the fungi are adapting. And we know that they can adapt because biotechnology companies, for example, that paper at the bottom there, they try to get some pest control insects to be more resistant to higher temperatures. Why? Because the insects are smart, and whenever they get infected, they sun themselves. So they're able to raise their temperature. And what you can see in that paper is that they could adapt a fungus that could grow only up to about 30 degrees to grow at 37 in about 6 months. The fungi can be selected for higher temperature. So we argue that global warming may bring new fungal diseases for mammals. And that there is a new organism, Candida auris, that has emerged for which there is really no good explanation. It turns out that it emerged at the same time in 3 continents, in South Africa, India, and Venezuela. The isolates between the 3 locations are genetically very distant. It's not like somebody brought it from Venezuela to India or vice versa. The organism is not known to medicine. And we have proposed that this may be the first example of a fungus that is adapted to higher temperatures to defeat the mammalian thermal barrier. So some evidence consistent with that has emerged. The organism has now been successfully isolated from the environment, the Andaman Islands, in 2 places. One was a beach frequented by humans. The organisms there were already temperature adapted to higher temperatures. However, the one that was recovered from a marsh was not as thermal tolerant. What it suggests is that the organisms in the environment may be already feeling the effects of climate change and adapting. Now, it's what you need to think is not the average global temperature, which may be 0.5 degree or 1 degree. It's the number of really hot days. Every hot day provides a selection event. And in fact, there is some circumstantial evidence for this that we published 6 years ago. This was again looking at the collection in the Netherlands. And what you see there are 4 curves. The top curves are the Ascomyces. These are organisms that are already thermotolerant, and you see there is not a lot of change. But the Basidiomyces are notorious for being less thermotolerant among the fungi. And the curves are trending upwards, suggesting that the Basidiomyces were adapting beginning in the late 1980s. Now, add to that human core temperatures have dropped in the last century. And we may be in a collision here. People are getting colder, fungi are learning to adapt to higher temperatures. This may be the result of a lot less infectious diseases today than there were in the past, since inflammation tends to raise body temperature. Can we predict future threats? Well, I got to take you on a little side trip. And I want you to think about the epistemological levels of knowledge. And let's look at the solar system first. You could look out into the night sky and you can stargaze. Then you can notice patterns, the constellation and the wanderers, the planets. Then theories get developed. We had the Ptolemaic theory for about 1400 years, and it was replaced by the Copernican theory that placed the sun at the center of the universe. Well, we know that that's not the case anymore, but that's just giving you a sense of the epistemological levels. Then you have mechanisms, you have gravitation, gravity, Newtonian physics, and now we have general relativity. But there is a deeper level that you need to know in order to know whether things are predictive or not. And that is, what are the dynamics of the system? So is the system deterministic? Does it work like clockwork? Is it chaotic? And I'll tell you more about chaos in a minute. Or is it completely random? Well, it turns out that our solar system is chaotic. We cannot predict the future with certainty. There is about a 1/3 chance that Mercury is going to be kicked out of its orbit and will wander through the interstellar void in the future. We simply cannot predict it. In biology, in microbial pathogenesis, you could do the same thing. We could have observation. Is the host susceptible? You could have patterns, associate and correlate. In theory, well, we have at least the damage framework. I'm sure there are better theories out there, but at least we have one to work with. And mechanisms where a lot of people work with, virulence factors. But to know the future, to be able to predict, you need to know the dynamics of the system. And very little work has been done in this. It is an interest in my lab. So I want you to bear with me. I'm going to take you through some math, but I promise to try to make this understandable. First, I got to tell you about Cryptococcus neoformans, because that's going to be the organism that we are going to be using to probe the dynamics of infectious diseases. So it is a so-called opportunistic pathogen. Most people are infected, but disease is very rare unless the host becomes immunosuppressed. Strangely, it has a very sophisticated virulent strategy, raising the question, why should an organism that lives in the soil have a sophisticated animal virulent strategy? And second, one can use it to induce disease in very different hosts, from flies to plants. Think about that's non-specificity. Why should a soil organism with no need for any of these hosts manage to subvert such diverse host defenses? Well, the answer to the first one emerged a couple of decades ago, and that was that apparent Cryptococcus, like all the soil organisms and water organisms, learns to be virulent from the fact that it must protect itself against amoeba. These organisms are being under constant predation. So when they get into the lung and they run into a macrophage, they use the same strategy they use to protect themselves against amoeba to protect themselves against macrophages. Often the pathogenic strategy is very similar. And how do they manage to cause disease against such very different systems? Again, think of amoeboid predators in the soil. There are thousands of amoeba species. Their major threat are eukaryotes. So what they do is they hit eukaryotes at common points. They don't know what's going to eat them, but they know that they have mitochondria and ribosomes. So they tend to interfere with common pathways. They achieve non-specificity by targeting the nodes. We know that the cryptococcal pathogenic strategy dates to the Cretaceous. How can we possibly know that? Well, you take strains that are separated by 100 million years. You take them into the lab, you give them to macrophages, and you find that they are virtually indistinguishable. This strategy was arrived a long time ago and probably honed and maintained by interactions with amoeboid predators. We know where it emerged in Pangaea. Pangaea was the last great supercontinent when all the continents get together. So we know we were, by molecular mapping, proposed that it came from that region where South America and Africa interacted. And to give you a sense of the power of ideas, because this is nothing more than an idea, other groups have taken that idea and then mapped cryptococcal strains across the planet. And they have been able to show, for example, and explain why isolates from Spain are close to those from Eurasia. Because at the time, when the continents broke up, these were close to each other. So words about dynamical systems. So in one slide, I'm going to tell you a lot about dynamical systems. So all dynamical systems, that is the mathematics that involves systems that change, it's either deterministic or stochastic. A deterministic system is clockwork. You set it up and it keeps going. It's a system where there is no randomness. Now, here is the complication where I always tell you, chaotic systems are deterministic. Chaos in mathematics doesn't mean chaos in regular life. Chaos in mathematics is not a messy room. Chaos in mathematics means that the system is so susceptible to getting the initial conditions right that very small changes propagate through time to give you very different outcomes. But if you could go back and set up the weather of the Earth exactly as it was in the year 1900, you would get the same weather pattern through the entire century. Or they can be a stochastic. Stochastic systems are random. They're inherently unpredictable. For example, you can have a radioactive atom suspended in a laser beam, for example, or hold it by some way. And there is no power in the universe that's going to tell you what it's going to decay in the next minute or at the end of time. The important thing is that because of chaos, even though it's deterministic, it's unpredictable. So chaos systems are unpredictable into the future, and so are stochastic. So chaos theory is a new theory in mathematics, and the example is that it does a butterfly beating its wings in Brazil set up a tornado in Texas. Do small changes propagate through complex systems? Detecting chaos, we're not going to get into the mathematics. All I will tell you is that there are ways of doing it. You can take the data that is generated in the lab, and you could do what is known as an ordinal pattern analysis that allows you to differentiate your system into a stochastic versus deterministic. And then you can do what is called a power spectrum analysis that allows it to break it into chaotic versus periodic. And let's just walk through some of this. So here is the butterfly beating its wings in Brazil. So you want to know whether the system is a stochastic or chaotic. Well, if it is a stochastic, you will find the butterfly wings at every position as a function of time. If it is chaotic, you're going to be missing some positions. And those positions show themselves what are known as forbidden patterns or forbidden ordinals. They're simply not there. So let's look at chance in the phagot lysosome. So the pH in the phagot lysosome is a well-known cellular biological effect. All phagosomes acidify. And this is being studied for half a century. So what we look at the question is, does the cell know where a phagosome final endpoint in pH is? And we looked at with beads and we looked with Cryptococcus. So when you look with beads, what you find is no ordinal patterns. All the pH values are represented. What it suggests is that acidification is a stochastic process. Cell doesn't know what the final pH is going to be. It's probably regulated by the number of pumps, the size of the phagosome, in a lot of very small things that all combine to give you a beautiful Gaussian distribution. The pH distributions for beads approximate normality. It's a stochastic system. Throw Cryptococcus in, and it pushes it away from normality. Cryptococcus disrupts the dynamic system in order to better survive. Many pathogenic microbes do this. Tuberculosis does this. Others poke hole in the membranes. Others mess with acidification. Cryptococcus messes with acidification. It secretes urease that converts urea into ammonia. The capsule enlargement dilutes the volume of the phagosome. Phospholipase damages the membrane and allows cytoplasmic components to come in and acid to leak out. And then you have glucuronic groups in the capsule that buffer the pH. So what you're seeing is biological bed hedging at the level of the phagosome. Biology uses bed hedging to survive, for species to survive. Birds lay different size eggs. They don't know whether the coming summer is going to be a good year or a bad year. And the way that a small egg does better in a bad year, a good egg does better in a good year. So what Cryptococcus does is it interferes with bed hedging by moving the conditions to a situation of optimal pH. So what you're seeing here is that the fundamental interaction between a microbe and the host cell. This is very reductionist. the interaction is unpredictable. Because even though the host cell tries to optimize things, it doesn't know what it's going to eat. So it optimizes the pH to hedge his heads, to hedge his beds, I'm sorry. What you instead see is that the microbes mess with that. What about chaos? Well, it's a much more difficult problem. We took an early peek into chaos about 10 years ago, and we did not find it. But not finding it doesn't mean it's there. It just means that whatever methods we use were not revealing for it. I can tell you that work has continued. We are in the midst of writing up a paper, again with my friend Abid Bergman, my collaborator Nicole Broderick, and others, in which we have looked at 3 systems: at C. elegans, Drosophila, and moths, infected with 3 different pathogens. And what you see now is, when you look at the moths, you see forbidden intervals. You see evidence of chaos. So what you're seeing is the interaction at what I show you, the phagosome, the stochastic interactions, fundamentally unpredictable. You're seeing at the level of the organisms, traces or hints of chaotic dynamics, also unpredictable. What does that mean? Well, it's going to be very difficult to predict what microbes are pathogenic. Virulence factors manifest new emergent properties that could not be easily predicted from their known roles. Urease is a nutritional enzyme. It's used to obtain nitrogen. However, in the phagosome, it generates ammonia, and it basically causes a new condition that would have been very difficult to predict. You can explain it once you see it, but predicting it from first principles is very difficult. Host cell interactions in the phagosome are stochastic, implying randomness in final outcomes, and host microbe interactions show chaotic signatures. But don't despair. Why not? Well, the ultimate chaotic system is the weather, and we have gotten really good at a weather prediction. In 2000, a 10-day weather forecast wasn't maybe 30% effective. Since then, we are almost up to 50%. What is different? It's not the dynamics in the system, but we have learned from experience and from mathematical modeling. So one is able then to, with the computing power, to get some degree of predictability. So probabilistic prediction may be possible in infectious diseases. And lastly, I want to tell you what happened with my lab. My lab learned to use these programs, and in the summer of 18, unknown to me, began to study my visits to the lab. Let's say Arturo walks in at 9, and then he walks in at 9:30 and doesn't return to 2 o'clock. So the first interval was 30 minutes. The second interval was 4.5 hours. And what they did is they all got together and they mapped this for a whole summer. And they were able to show that in my visits to the lab, which is a dynamical system, there are forbidden intervals. That means that it shows evidence of chaos. I was so impressed with that. I was so impressed. Sat there and really, one of those moments in which you really enjoy seeing your trainees do much better than you do. I never had the idea that they could do this. And then I began to worry that they had done human research without IRB approval. And I immediately, after I left the lab, I went to my office. I called the dean and I said, look, this cannot be discouraged. This is amazing work. And there was silence at the other end. And the person said to me, look, it's not research unless you plan to publish it. So it's okay. But if you plan to publish any of this, then you need IRB approval, retroactive IRB approval. And I left, we're not going to publish this, but it gives you a sense of the power of this way of thinking, that people signing in and signing out of the Zoom, people, the way they interact, these dynamical systems, if we understood them, we may have a much better handle on things. So in closing thoughts, the thermal barrier that protects mammals is narrowing as the world is getting warmer. The fungi are adapting to higher temperatures. Humans are getting colder. I think that means trouble ahead. When you think of global warming, focus on the number of very hot days, not on the average temperature. every hot day provides an event for fungal adaptation and selection. New fungal diseases are predicted to emerge. Candida auris may have been the first, the proverbial canary in the coal mine. So in your research and in your life, whatever you do, consider investigating the variable of temperature because it is and will be important. And with that, thank you for having me. Thank you for letting me be part of your 200-year celebration. It is a great honor. I only wish I was there with you to celebrate in person. Thank you.
Is this on? Thank you for that, Arturo. There was massive applause in the room here, and I hope you can hear us through Zoom right now. I'm not sure how the links are doing.
I can hear you. Can you?
Great. Okay. So Don is on now, and he's monitoring the online questions, and I think he has a few questions of his own as well. So Don, I'm going to let you take it away and ask some questions from yourself and also those that have been sent to you.
Yeah, I'd like to remind everybody watching this on YouTube, the questions, there's a link under the YouTube video document, so you can link and type in, as some have already done, forward those on to Arturo. First question, the fungal bloom at the Permian era is an evolutionary gate that organisms had to face. Given that fungi remove pathogens of, why did plants not evolve resistant to fungi as mammals did?
Well, they are resistant. The entire world is covered. It's just that they're more susceptible. So diseases of plants, and they are very rare diseases of mammals. That we live in a green planet. There is water. It's a test fact that plants are just not as resistant. I want to say that when I have a chance to watch myself give it, and I sat here for minutes cringing, I get standing up, and I did the way how I plan to learn from this, and you can be sure if I won't try to get you seized by the constant back and forth. What an experience!
Yes, watching themselves on the reasons. We
oh God! Yeah, I thought that when you turn it, the show would be the camera. I didn't think I wouldn't be forth. But next time I will be sitting, possibly tie my hands to make sure that I don't have any of this interference.
Other question for you. Would we predict fungi undergoing thermal would migrate through the framework of colonized opportunists before reaching, using the dreaded word here? And therefore, should we be monitoring the migrant for early new invaders and new organisms of concern?
So the answer is, in fact, before in the last few 2019, I was working with collaborators to try to do the matrix, because it turns out that there is a lot of it there. There are, we know that the temperature is 35 degrees. Some of these are pathogenic and to ectothermic animals. Place them in a category that cannot go above 25 degrees. That work came to, some of you may know that I've been with convalescent class almost, but we want to hope to try to the matrix to see what we should be worried about already close to us, and that's how it.
There was a note in the chat to turn off our camera, not receiving our audio. Can you, are you able to?
Yeah. Okay, hopefully this works better. It feels like the internet is run on some dial-up modem from 1993, and we'll see if the Q&A goes better with the two of you just on audio. Don, did you have any more questions?
One last question, comment, and is, would the potential concept was discussed, factors under the factor that drive to prescribe antibiotics or viruses? That's more a comment. You have a thought on that, Arturo, but that was thrown out.
I think that more of a comment than it's something to worry about, and this is misuse of antibiotics. I think is more sociological. It is very hard, and a physician takes care, goes into their office, waiting for a long time, and be able to do something, and feel like, well, drugs are harmless, so prescribe it. The fact is that they have in the microbiome and all that, and I think more people want to do something and are thinking about potential. But I urge all that to be antibiotics. They cost on the individual from super infection that to tremendous cost to society in the form of systems.
Okay.
That's it for me, Marcel.
Okay, thank you. Arturo, I'd just like to point out that now that we have a still picture of you, you're not moving around as much. So this is all good. We have a question at table 8.
Arturo, I thought that was one of the most fascinating talks I've ever heard. But I just wanted to ask you, or maybe push the questions a little bit further. And I get it through the evolutionary history that fungi have emerged. But what allows for the overwhelming preferential involvement of ascomycetes over basidiomycetes or other mycetes, if in fact, we're talking about, a sort of a general filter?
And Arturo, that was a question from Don Vin, in case you didn't hear from the back.
So the ask, that is the Candida, plasma, and all that group of organisms is much more adapted. They grow much easier than mycetes. Mice is interesting, are more found in the areas of the planet. So I think what stochastic, I think that you more ascomycetes that are already temperature, and some of them happen to be sweet. So most of our fungi for humans are Cryptococcus is the oddball out mycetes to have temperature. It's one that can grow at 30, therefore is pathogenic. But I think we need to, I think that all these organisms and thing that we tend to do is we always tend to prepare for and learning for birth the last 30 years, and it's coronavirus that society is a part in. I think the fun to watch.
Is there one more question? Maybe you can introduce yourself to Arturo as well, because I'm not sure if he can see people's names from the microphone.
Thank you. I'm Leonardo Jurado, a PhD student in McGill University pathology program. Thank you. Talking about like the evolution of potential, the pathogenicity and virulence. I've been working on TV during my last 5 years probably. Right now I'm working on cancer, but it doesn't matter. But my question is, when a bacteria such as Mycobacterium tuberculosis decide to become like a latent pathogen instead of like produce or generate immediately like an infectious disease, like acute infectious disease, like when in the evolutionary I don't know how to say that, such really pathogenic bacteria decided to become like latent instead of trigger like acute infectious disease? Thank you.
So Leonardo, I work on TV. I want you to think from now on. I don't want you to be decides to do anything. Tuberculosis became associated with sometime Neolithic times. It's cause selection in human, and we have cause selection. The interaction, you know, for most tuberculosis is genic. The majority become PPD positive. It doesn't cause disease. But tuberculosis is one of the few organisms that needs to cause disease in order to pass. So it causes disease in the lungs, people to cough, causes it to pass itself. So it's under selection. The humans who get it. The direction of to the future is unknown. Don't know if it is heading influence or we're heading for more virulence. I think kind of things that are infections, but we don't have a handle on. Most people tend to think initiation leads virulence. That is, over time, organisms evolve to be less virulent. That is only the organism can pass from host to host without disease. It has disease in some way. There is no virulence.
Okay. So Arturo, I'm going to wrap up now, in part because my understanding is that you and Don hear each other great, and we here in our room don't hear you very well, which tells us that the principal of the university who's in the room is going to fix things. And the next time we have a meeting here in the faculty club, everything is going to be working great. I will defer to another day to ask you whether not only humans are at threat, but bananas and other fungal diseases that we should all be worried about, and just tell you that we really appreciate your thought-provoking talk. We really appreciated your reflections that thought is somewhat underrated and underpublished, and we look forward to having you come in person at some point in the future and sharing with us the updated version of this talk and where we're headed with global warming, among other challenges. So thank you very much, Arturo. Don, do you have any last thoughts, or thank you as well to share?
Bye, thank you.
Yeah.
It was, I'm glad you paid me.
Okay. And on that note, we still have technical problems at this end, but I'm going to call up the co-director of MI4, Marie Hudson, to give some closing remarks. So thank you again, Don, and thank you again, Arturo.
Alors, thank you. Bonsoir, tout le monde. First of all, I'd like to extend my thanks as well to Dr. Casadevall. When I met Dr. Casadevall about 10 days ago, he said, I hope that there will be something in my talk to interest even a rheumatologist. And I would say that that was quite the understatement. Bringing it a little bit more concretely, though, I really very much appreciated your bringing to the fore the fact that some of our science, some of what we do every day, lacks some theory. I think that theory gets lost in the practicalities of day-to-day research, but I think it's such an important message, and that's the message I'm going to take home as a rheumatologist and as a scientist tonight, and I hope that some of my colleagues do that as well. Don, on a personal note, I hope you're still there. I've always known that you were a fun guy, but I never really knew what you were exactly doing. But after this talk tonight, I have a whole new appreciation. Keep up the good work. And on a personal note, best of luck with your new endeavors. We look forward to having you back at McGill soon. So while we're at the thank yous, I would like to say a few other thank yous before moving on. Principal Fortier, Vice Principal Craigo, you have been our steadfast supporters at MI4 since inception. We thank you tremendously for that support. We thank you for being here tonight. Your presence is really strong encouragement as we move forward with our next challenges at MI4. So thank you for being here. The other people I would like to thank are the people who really make us look good, because we stand here and everything generally looks easy, but we have an amazing staff of people that are there, and it is that staff who really is behind what works at MI4. I would like to point them out. So Kendra Tonkin, who's our program manager, Tasha Allende, who's our... Tasha Allende, who is our advisor par excellence, Lynn Derry, who is our fabulous bookkeeper. Yeah, Lynn, thank you. And the new kid on the block is Jean-Etienne Sheehy, who is our communications expert, who's really trying to help us with getting the message from MI4 out there, and we strongly believe that he will help us do that. I'd also like to thank the donors. I think that we have been privileged at MI4 to have such support from the donors. And the way I look at the donors at MI4 is that these are not just any donors. These are donors invested in science. And I appreciate those donors. And I look forward to meeting more and hopefully moving forward with many of them. And finally, I would like to thank Don and Marcel on behalf of the MI4 community. MI4 is your brainchild, and I think your brainchild is thriving. We as a community thank you, and as we move forward with the next few years, I hope that we will make you proud. So thank you on behalf of the group. So I have a few updates about MI4. One of the things that we're not doing this year that we've done in previous years that I thought was very useful was to take the opportunity to give you an official update on our activities. We have not had the time to do that this year. However, we have worked quite hard at putting together a very nice annual report. The annual report has lots of specifics and details. It is ready. It is at the production phase, so la mise en page and so on. So we look forward to sharing the annual report where we will be showing you everything that we as a community have been busy at doing in the last year. Looking forward to the next year, we're really looking forward to the 4th round of our seed fund grants. The competition just closed about a week or so ago. We had 18 applications, which is 2 more than last year, which is really exciting. So we look forward to reviewing those grants, and we hope to have the results available by March. We have a MI4 Ludmer competition that is still active. That closes on December 8th, so please have a look at that if you're interested in applying for that. And obviously, we're still open in terms of our flagship funding and our co-funding opportunities. So bring us your best ideas, because that's what we're here for, trying to move things forward. One of the most exciting things I think that's going to happen to us next year, it's also a little bit of a challenge, is we have worked hard at putting together an external advisory board of international experts who have agreed to review our program and to help us understand what works and what we should be doing better. So we haven't had a chance to post the board members yet on the website, but those should be done shortly, so you'll be able to see who's going to be on the board. And I think you're going to agree that we've really been lucked out and had been able to engage some people from around the world that are going to be very helpful. So we have our first board introductory meeting next week, and then the official review will happen sometime in the new year. And then the last thing that I was really excited about announcing tonight, but I think I'm going to take a step back, is we were seriously thinking of bringing back our in-person Dragon Dens events, which many of you have attended and that are very exciting, very important for our networking. But with the Omicron variant coming along, I think we're just going to put that back on the back burner. But it's definitely within our priorities to try to get back together and start to network and cross-fertilize and work together. So that ends the official part of this session. We're going to move on to the next part of the session where we're going to allow participants to exchange a little bit. We encourage you during this part. Most of you are supposed to stay seated. You're allowed to go get a drink. When you do so, you have to wear your mask. When you are at your tables, you're encouraged to wear a mask unless you're eating. But we have identified a few MI4 ambassadors who have the privilege of going from table to table, hoping that some new people can meet each other and can discover what some of the exciting science that we're doing. So these MI4 ambassadors are Don Vin, so you'll be seeing that some of these people are going to be moving around. Sonia Del Rincon, Judith Mandel, Geneviève Genet, Jessie Shapiro, and April Rose. So there's not a lot of time left. I don't think they'll be able to go at every table, but hopefully they'll be able to go at a few tables. They can introduce themselves, tell you a little bit about themselves, and then you can ask some questions. So I think that that's it. So with that, I thank you for coming out tonight. I wish you a good rest of Infection and Immunity Week. And we look forward to more from the McGill's Bicentennial.